This is our second video on proteins, and in this one I'll be talking about the 4 levels of protein structure and the learning objectives. The things I want you to be able to do after watching this video is I want you to be able to describe the importance of weak interactions in protein structure, to describe the elements of a polypeptide chain necessary for each level of protein structure. I want you to be able to differentiate between the 4 levels of protein structure, compare and contrast the 2 forms of secondary structures, and illustrate the effect of changes in pH on protein structure. Okay, so let's start with the first and most fundamental level of protein structure. We call that the primary structure. And essentially, if you watched the previous video, you know what the primary structure is. It is simply the sequence of amino acids that are connected to one another via peptide bonds. So in this image here, looking at all the amino acids that are connected to one another in this chain, seeing the specific sequence of amino acids within this chain, we are looking then at the primary structure. Okay, and this protein happens to be transthyretin. Right, we know something about the primary structure. We know that this chain has an amino terminal end. We know it has a carboxy terminal end. We know that the R groups of each one of these amino acids are sticking out, right? So here we have all the bonds keeping all of the amino acids together, and here we have the R groups. So that's it. The primary structure is defined as the sequence of amino acids connected to one another by peptide bonds. That's it. Next, the secondary structure of proteins now takes into account electronegative oxygens and partially positively charged hydrogens within the backbone of that polypeptide. So again, we're ignoring the R groups. In fact, all I've got written here is R for the R group. R, 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 R. Right? So those can be anything. What we're looking at is the part of the polypeptide chain that is identical in every protein. Okay, you can kind of read it like NCC, NCC. What you can see is this oxygen here, this carbonyl oxygen, electronegative. This hydrogen here connected to that nitrogen, that is partially positively charged. So these two make a great hydrogen bonding pair. And what happens is this polypeptide chain folds in very specific ways to accommodate as much hydrogen bonding between these hydrogens and these oxygens as possible. And there's several different types of secondary structure, only 2 of which we are going to focus on in this course. The first is alpha helices. And what they are is the chain, the polypeptide chain forms this helix, right? This twisting of that chain around a central axis. And what it does is it maximizes hydrogen bonds between those hydrogens and those oxygens I was talking about. But what's important to note is that it's not the hydrogen and oxygen of 2 adjacent amino acids. Because of this twisting, those 2 amino acids that are involved in that hydrogen bond are about 4 amino acids apart from each other. So we've got a hydrogen bond there, one there, another there, another there. And so this maximizing of hydrogen bonds with this helical structure is called an alpha helix. Another type of secondary structure is called the beta pleated sheet. And what you have is you get a long chain here. Whoops, come back here. And then the chain loops back on itself, and across those chains, we have hydrogen bonds. And now these could be separate polypeptide chains of two different proteins, or more commonly, the same protein that's looping back and forth on top of itself. And again, it's that same electronegative oxygen, or that partially negative oxygen, that partially positive hydrogen, but they are now, this time, unlike the alpha helix, we are many amino acids apart from each other. The amino acid here is many amino acids away from the one right here. Okay, those are called beta pleated sheets. And notice the R groups aren't involved. The R groups are up here. There's an R group. Here the R groups are sticking out of that helix. Those R groups are important in the next level of protein structure, and that's the tertiary structure. So now this third level takes advantage of the R groups of the amino acids within that chain. So we've got R groups that are polar, and those can form hydrogen bonds. We've got R groups that are ionic, right? Here's an amino group of within an R group. Here's a carboxyl group within an R group. Look, they form a nice ionic bond there. We have hydrophobic interactions. So these non-polar R groups are not going to be able to interact with water, and they're going to sort of clump into the center of the protein. The protein is going to kind of fold around those hydrophobic amino acids, or R groups. And then, remember, we have that special interaction that occurs between adjacent sulfhydryl groups, forming this disulfide bridge. Now remember, this isn't a weak interaction. This is a covalent bond that forms there. So this protein then is going to fold to accommodate all of these various weak interactions. And it's not random, right? It folds in a very specific way every time. And by the end of it, you now have this protein where you can see all three elements of protein structure. You can see the primary structure in that, well, it's just a sequence of amino acids connected to one another via peptide bonds. You can see elements of an alpha helix, the secondary structure. There's an alpha helix there. These guys here are all beta pleated sheets. And then, of course, how all of those secondary elements kind of fold over on top of each other, right? That is due to the tertiary structure. So all these structures, the primary, the secondary, the tertiary, they exist simultaneously to form a properly folded protein. And it's only when that protein is folded correctly does it have the correct function. If you do something to unfold this protein, it will no longer have the proper function. Now, there are other proteins that have one higher level called the quaternary level of protein structure. Now, quaternary proteins are proteins that have more than one polypeptide chain. So for example, collagen has 1, 2, 3 polypeptide chains. Hemoglobin has 1, 2, 3, 4 polypeptide chains. Now these polypeptide chains have to come together in very specific ways, and they do so not via covalent bonds, okay, but via these weak interactions. And we call these type of proteins oligomers. They are proteins that are made up of more than one polypeptide chain. Collagens are trimers because they have 1, 2, 3 chains, and hemoglobin is a tetramer because it has 1, 2, 3, 4 chains. Okay, so now we're going to get back to something we discussed in that first lecture, and that is this. The final structure of a protein determines its function. If it doesn't have the right structure, it's not going to have the right function. Okay, so the quaternary is dependent on the tertiary is dependent on the secondary, and the secondary is dependent upon the primary structure of amino acids. So it's the sequence of amino acids that are connected via peptide bonds that determine the secondary, the tertiary, and if possible, the quaternary structure of that protein and therefore the function. So we can make an important concept here. The primary structure of a protein is what determines the function of that protein. If you mess up the sequence of amino acids within within the polypeptide, within the protein, you're going to mess up the final structure and therefore the final function. That's a really important concept to understand, especially when we begin talking a little bit more detail about molecular evolution and evolution in general. Now, what that also means is that the only information necessary for a protein to properly fold is right here, that primary structure. If you take a protein and you unfold that protein, it will fold back together if you give it the proper conditions. It doesn't need any other information other than having the proper sequence of amino acids. Okay? With that said, however, we have a problem that proteins are made within a cell, and in that cell there's lots of other proteins. So while that protein is trying to fold, it sometimes gets tangled up with other proteins. And so there's a special class of molecules, they themselves are proteins, called chaperonins. And what they do is they allow a nice isolated environment for a protein to go in, and now it's independent. It's isolated from all other proteins within the cell. It can fold properly on its own, and then it's released. So these chaperonins, they don't provide information for how a protein folds. They just provide an isolated environment so that the proteins don't accidentally get tangled up with other proteins as they are folding into their correct structure. Okay, proteins evolved under specific conditions. Okay, so as those proteins were evolving, right, that the organisms were evolving, that the proteins were in, they evolved to have a proper function, proper structure under a specific pH, a specific temperature, specific ionic conditions, etc., etc. If you take a protein and you put it into an environment that strays away from those optimal conditions, that protein is going to lose its structure. The protein is going to lose its function. Why? Because when you put a protein into conditions that it did not evolve in, when you stray away from those optimal temperatures and pHs and ionic conditions, weak interactions are going to break, and that protein is going to denature. Okay? So here we have our normal protein, and here we put it into conditions that are not ideal for it. Maybe it's too hot, maybe it's too cold, maybe the pH is too high, maybe the pH is too low. That's going to break the weak interactions, and the protein unfolds, the protein denatures, the protein will no longer have its proper function. But this is not permanent. That's important. If you place this denature protein back into its optimal conditions, that being its optimal temperature, its optimal pH, its optimal ionic conditions, it will spontaneously renature into its normal structure, its native conformations. All right, so what I want to do is I want to show you, based on everything that you've learned about functional groups, about deprotonated and protonated, I want to show you how changes in pH can denature proteins. All right, so what you'll definitely want to do is make sure that you review the structure of carboxyl groups and amino groups and show how they can be protonated and deprotonated, because that's absolutely important for understanding what I'm about to show you here. So let's say we have a protein, and that protein, right, is a string of amino acids, And here's a fold. This protein is folding in a very specific way, and the reason it's folding like this is because it's accommodating a very specific ionic interaction. It is accommodating an interaction between this amino acid, O minus, and this one, NH3 plus. So this amino acid has a carboxyl group in its R group. This amino acid here has a amino group in its R group. So maybe this one is aspartate, maybe this is lysine. And notice that they form a really nice ionic interaction here. The positive charge of this amino group and the negative charge of that carboxyl group. And that's why this protein is folding the way it is. Now we know, let's see, I'll write it down here. Remember, if we take a carboxyl group, COO minus, and we add a proton, right, we can do this, COOH. This is an equilibrium. This is at an equilibrium. And remember that when we have a chemical reaction and equilibrium, what happens when we increase the concentration of one of the reactants? So now let's say we are increasing the concentration of protons. Let's write that right here. So the concentration is going up. What's another way of saying the concentration of protons? Right? The pH. So, in other words, what we're going to do to this solution that this protein is dissolved in is we are going to increase its hydrogen ion concentration, which means we are going to decrease its pH. Okay? We want to see, well, what effect does that have on this protein? So if we are increasing the hydrogen ion concentration, if we're decreasing the pH, what that means is we're adding more and more and more of these. We're adding more and more and more protons in our solution. Now, if we take a look here at this reaction, and we understand the principles of chemistry and equilibrium, if we increase the concentration of one of the reactants, that equilibrium is going to shift to the right, towards the products. That means we're going to end up having more of this in solution. So as we increase the concentration of protons, suddenly more and more of these molecules, that carboxyl group is going to be protonated. Now, adding more and more and more protons is not going to affect this amino group. It already has as many hydrogens it can hold, 3. It's already protonated, but this carboxyl group was not protonated. Now it is. Right? At that low pH, suddenly most of those carboxyl groups are protonated, and what happens? If we have protonated carboxyl group, we no longer have that charge, which means there's no longer an ionic interaction that's available here, and this fold will undo itself. It'll become unfolded, and now it doesn't function anymore properly. So here I illustrated to you, using all the easy principles we've already learned before, how pH, or changes in pH, right? We've really reduced the pH, can inhibit or denature a protein and stop it from functioning. Let's go the opposite way. Let's do this now. Instead, all right, we're now going to take our protons concentration, and now we are going to lower it. We're going to be removing protons, which means if we think about it in regards to pH, we are going to increase the pH. Okay, so little by little, what that means is this. Removing protons, right? We're lowering. We're adding base, right? We're lowering the pH, removing protons. removing protons. Eventually, right, we're going to drive this reaction back to the left here. We are going to, at some point, get to a situation we are back to the optimal pH. Fantastic. If we stop now, if we stop increasing the pH, if we stop lowering the hydrogen ion concentration, our protein renatures, and we're good to go. But let's keep going. Let's keep going. Let's keep adding more and more base. Let's keep driving up the pH, driving down the hydrogen ion concentration. And now what we're going to have to look at is something else. Now, what we're going to have to look at is this. NH3 plus is in equilibrium with NH2 plus H plus. And what we're doing now is we are driving the concentration of protons down. We keep adding base and base and base. So we're taking away protons, taking away protons, taking away protons. What will eventually happen as we take away, so if we remove product, our equilibrium moves to create more product, which means eventually what we're going to do is we're going to have more and more NH2 and less of NH3 plus. So eventually, as we keep reducing the hydrogen ion concentration, as we keep increasing the pH, eventually we're going to lose one of those 3 hydrogens. What happens? We also lose the charge, right? We don't have a charge there, meaning now that we've increased the pH so much, that amino group becomes deprotonated, it no longer has a charge, and we no longer have that ionic interaction possible. The protein unfolds, it denatures, and it no longer functions. So what we've seen here then is if you have a protein, it exists, right, it's evolved to a specific pH. And if you take that pH and bring it up too high, the protein will denature. If you take that pH and then you drive it too low, the protein will denature. It has an optimal pH at which the carboxyl group is negatively charged, the amino group is positively charged, and they can form that important ionic interaction. All right, so make sure you watch this a couple times. There's some really important stuff in this video. So go through each one of these learning objectives. Go through some of those demonstrations that I've done. Do them for yourself. See for yourself how they work. And then review some of the other videos. For example, the functional group video will help you as well.